Hello everyone, my name is Justin Ross, and I'm excited to be speaking to everyone today about Oxford Nanopore sequencing technology. So without further ado, let's jump right in. To begin, I will discuss how Oxford Nanopore sequencing technology works. Then I will discuss the sequencing workflow from start to finish, how to actually conduct one of these runs, and hopefully I'll give some practical, helpful tips along the way. Finally, I will discuss the data analysis pipeline that my lab has been doing, as well as some practical applications that you can use this technology for, including some examples from our own research. So, to begin, how does Oxford Nanopore sequencing actually work? So the nanopore, which is where ONT technology gets its name from, this nanopore is a protein pore through which molecules from your library moves, your DNA or RNA. A motor protein, which is an enzyme ligated to the ends of the DNA, has helicase activity, unzips, and pushes your molecule through the nanopore. And then finally, your nanopore is embedded within a membrane, which is a sheet with electrical current passing through. This current is disrupted when nucleotides are passing through it, and this disruption is ultimately recorded. All of this is embedded within microscaffolds, which stabilize and support the membrane and the nanopores. The sensor chip holds the nanopores and sends these electrical signals to the ASIC for processing. The ASIC is essentially a specialized microchip which controls the voltage going to the nanopores and then digitizes the electrical signal, which is recorded by the flow cell, which is the device into which the library is loaded, and the flow cell is plugged into the sequencer.
Hey, Justin.
The sequencer houses the flow cell and applies the current, measures the electrical disturbances, and uses this integrated basecalling software called Dorado to interpret the signal. So as I mentioned, when each nucleotide passes through the nanopore and the membrane, it produces this characteristic electrical squiggle, which is then interpreted as being whatever base it is by the sequencer. So in last slide, you saw the MinION. That's one example of a sequencer offered by ONT. There are a few other ones. For the purposes of this talk, I'm going to be discussing protocols that are for the Prometheon. That's the system that I've used at the SCIC, where I do all my sequencing. But it's just a matter of user preference for which sequencer actually is best for whoever's project. The MinION is really interesting. It's actually sort of pocket sized, so you can take it into the field if you'd like to. You can do sequencing pretty much anywhere at any time, but I've been using the Promethean. I've never used Illumina sequencing, but for those who have, I wanted to give a really brief comparison of the sort of the relative benefits of each. So nanopore sequencing is really good for ultra long reads, so up to 4 MB. It can do real-time sequencing, which is really nice because you can see how your data quality is and if everything is going well in real time. And it has relatively high accuracy. So I think the latest kits offer over 99% accuracy if you're above a certain quality threshold. So if you're above Q20, and I'll go into a little bit of what that means later, but relatively high accuracy. On the Illumina end of things, these are best for shorter reads. So I think it's like 10 to 500 base pair reads. It's a little bit more common. It's been around for longer, so there may be more documentation than exists for nanopore sequencing, and it has really, really high accuracy, near perfect modal accuracy. As I mentioned, there may be more protocol options available for Illumina, but there are still a ton of sequencing protocols and documentation provided by ONT. So if you want to do sequencing from plant tissue, animal tissue, if you want to do direct RNA, PCR amplified RNA, genomic DNA, there is probably an option that you can find. But again, I'm here to talk about my experiences with ONT sequencing. So for the purposes of this talk, I'll be going over just 2 protocols. The first being human variation sequencing from 10 kb extracted cell line samples. So this is the whole genome sequencing, and then direct RNA sequencing. The purposes of our experiments, we didn't want PCR bias or anything. So these are the two that I'll be focusing on today. So with that, we can jump into the sequencing workflow. So the first thing, of course, you need to do is from your sample, extract your DNA or RNA. I'm sure that many of you have done this at this point, so I'll try to be brief going over that. Then you prepare your library, and then you actually do the sequencing, and then, of course, finishing your run. Crucially, a huge component of this that I've found extremely necessary is making sure to check the quality of your samples between steps and even within the library prep protocol. Some critical QC factors to consider are fragment size. So if you're doing whole genome sequencing and you're looking for these ultra long reads, you want to be making sure that your fragment sizes are to your expectation. Same for RNA, making sure that your fragment size is in line with high quality, not ultra fragmented molecules. Concentration of your sample, if your concentration is too low, then you're probably not going to get robust output. And then sample purity, which I'll talk about a little bit later. I'll go over a couple instruments that I like to use for my quality control and my quality checking. All of these are available either through the Genomics Core or the SCIC. It's really easy to submit your sample for a relatively low cost if you'd like to check the quality of your sample. So first is the qubit. I'm sure many of you have heard about this instrument before. You can check DNA and RNA integrity and quantity. So here you see this RNA integrity and quality number here. 7.1 is on the lower end of what I would ideally sequence. It's a 0 to 10 scale, where 10 is like a perfectly non-degraded sample of RNA. 7 is sort of on the lower end here, so I don't believe that I went through sequencing this sample, but it's really nice because the qubit can tell you the breakdown of small and large RNA in your sample. The fragment analyzer is really nice as well because it actually gives you an idea of the exact sizes of the fragments and the abundance of those fragments in your sample, as well as the integrity measured through genomic quality number and RNA quality number, as well as the quantity or concentration of your sample. And finally, the nanodrop is really good for measuring purity of your sample and as well as the concentration. Okay, so with that, let's talk about sample extraction. I've personally extracted from both tissue samples and plates of cells. I'll be brief, but I did want to give a few tips for each of these sample types. So tips for extracting DNA, you want to make sure that you're minimizing the amount of like vortexing or really like rough shaking that you're doing with your DNA, as this can lead to excess fragmentation. That might be good depending on what you want, but in general, it's best to do this in a controlled way. Dry ethanol completely. So this is what I was talking about with the purity component with these samples. If you're doing like a DNA extraction that has some sort of like an ethanol component, you want to make sure that you allow the ethanol to dry completely, as well as like other contaminants you want to purify out, as these could interfere with the kit chemistry like later in like library prep. And then finally, this also goes for RNA, but in general, it's best to elute in smaller volumes because, of course, it's much easier to dilute your sample if it's too concentrated than to re-elute and try to shoot for more concentrated samples. Some tips for extracting RNA from samples. Just proper RNA like handling technique is really important here, especially with this like library prep protocol for ONT sequencing. You want to prevent RNase contamination to the best of your ability. We use RNase zap, super easy reagent, just spray on the benchtop, spray on the hands. Work over ice when possible, and aliquot to reduce freeze thaws, as these things can degrade your sample and lead you to lower data quality. And finally, and probably most importantly, tips for extracting from tissue samples. I know from experience, it's no fun trying to extract RNA, especially from coagulated blood samples. So use anticoagulants and preservatives when you're storing your samples as necessary, especially when you're working with tougher samples, like physically tougher samples. Ensure complete sample lysis, or else you're probably not going to get great yield from your samples. And in general, newer samples are usually better. Although if stored properly, you can probably keep some in the freezer for a while. But trying to extract from really old samples, generally not as good of an idea as extracting from newer samples. Okay, with that, let's discuss the DNA library prep protocol that I previously introduced. Step one that I always do when I'm prepping a DNA library is to shear your DNA. So ONT is really great for like these ultra long reads, but I found that for my purposes, I only really need like 10 kb long fragments. You can go longer. Usually when I do my DNA extraction, at least from cells, I get like 30 kb fragments, but these have the tendency to clog up your pores, and sort of counterintuitively, because of this potential for clogging, I find that shorter fragments might actually give you a larger N50 in the end. So your read lengths might actually end up longer if you shear down your DNA a little bit more. I didn't have access to a megaruptor for this, so I use Covair's G tubes, which are just these really easy disposable tubes. They shear the DNA through centrifugation, and depending on the speed at which you centrifuge, you can sort of pick which length you want for your DNA. Next, you go through DNA repair and end prep. And all of these, at least the second and third steps here, are as easy as purchasing a kit from ONT. They give you all the reagents. There may be a few third-party reagents you need to purchase. And then you just follow the steps throughout the ONT protocol. But the broad picture for this step is you prepare the DNA ends for the attachment to the adapters, which I mentioned earlier on in this presentation, for ligation to for the motor proteins, which is the third step. So the ligation of the adapters to the repaired DNA, and then cleaning up all of the shorter fragments that might be left over in your library. And from front to end, not including all of the quality checking, I would say that this entire protocol takes about an hour and a half to 2 hours, a little bit less if you're practiced, which I think is really incredible that this technology allows you to go from a plate of cells to a fully prepped library ready for sequencing in just a handful of hours. For the RNA library prep side, it's fairly similar, although instead of shearing, I actually do poly-A tailing. This step is also optional. If you're only interested with like the naturally poly-A molecules, then you don't have to do this step, although your output is likely going to be much lower. So I polyadenylate my RNA like manually. Then is the reverse transcription step. Again, this step is included in the protocol. This is where cDNA strands are synthesized for the stability of the molecule throughout the rest of the library prep phase. And then finally, the same thing, adapter ligation and cleanup. So this protocol probably takes a little bit longer. It's a little bit more finicky in my experience, but again, if you're practiced, this should only take you about 2.5 hours, not including the quality control. So now you have extracted your sample, you've prepped your library, and you are ready to do your sequencing run. The first step here is you want to make sure you check your flow cell to ensure that it has enough nanopores. I've found that most of the time when I get my Prometheon flow cell, it has like upwards of 6,000 pores. Although ONT guarantees that at least the Promethean flow cell should have 5,000 pores or more. If they have lower than that when you take them out of the box, then you can send them back for a new flow cell as long as it's within a certain time period of reception. Importantly, when you're evaluating if your flow cell has enough pores for your run, you want to keep your output target in mind. If you're only going for like a 1x coverage run and like a whole genomic sequencing run, then it might be okay for you to only have like 1.5 thousand pores in your flow cell. But if you want to get like 30x coverage, you might be shooting for more of like a 4 or 5,000 pore flow cell. So if you have the option, if you have multiple flow cells that you're choosing from which one to load your library into, keep output target in mind. Next, you want to prime your flow cell so it can be loaded with your library. Again, I'm hoping that folks here, if they're interested in using this technology in the future, can learn from my mistakes. You want to make sure that you load at a 90-degree angle so that you, to the best of your ability, avoid introducing air bubbles into your flow cell. So this is what it looks like to load. There's a little inlet port that you just load through a 1,000 microliter pipette into the flow cell. If you get air bubbles into your pipette, or sorry, into your flow cell, it's likely that your pores are going to dry out. And if that happens, it's very, very difficult to recover them. And you've effectively just spent part, if not all, of your flow cell just unloading without getting any data. And finally, you want to set up and start your run. So I do this at the SCIC, which is on the 10th floor of KCBD where the sequencer is. And the runs have customizable parameters. So after you load your library into a flow cell, plug it into the sequencer, all you have to do is fire up the MinION software on the computer, and you can customize your parameters to tailor your individual sequencing needs. So for example, if you are interested in getting methylation information, you can do that for your whole genome runs. If you're not interested in that, you can uncheck that box. If you're interested in only getting like a set amount of data, then that's totally fine. You can set that, and the sequencing run will actually stop after you reach that point. Same thing with time. You can stop it or you can set it so it stops at a certain time after your sequencing run begins. So there are a lot of these options. They're sort of contingent upon what the user wants. While your sample is running, there are a few things that I would recommend checking for. As I've had is where I load a library, start the sequencing run, and then I noticed that due to the real-time base calling, I can see, okay, the data is not looking so good. I should probably stop my run and reload the sample or use a different flow cell or something like that. First thing you should check for is sequencing speed. If you're getting like a really low sequencing readout in the first like hour after sequencing, it's probably unlikely that it's going to improve over time. So they provide these really nice graphs and charts so that you can tell this in real time. Here you see the total number of reads expressed in this blue line. The green line is the past reads. This is based on the quality score of your reads. Red is failed. And then this gray line, which peaks at the end of the run, is the skipped reads, basically the reads that due to computational reasons, the sequencer just cannot read in real time. But you get a really good sense, even if it isn't like perfectly every single read is base called in real time, you get a really good sense of what's going on. Next thing you want to check for is, of course, pore loss. I've had experiences where I load the library improperly, and within the first hour of running it, my pore count just completely craters. Again, it's unlikely that this is going to get better the longer you wait, so you might want to take your flow cell out. In general, you can expect maybe it depends on the sample that you're running and the goals for your data output, like the amount of time that you're running a sample, but you can expect like a 30% pore decrease, I'd say, generally after a completed run. You want to check you want to check your Q score. So as well, so you get this nice histogram while you're sequencing that tells you the quality of each read and the abundance of those reads at each quality point. Sometimes, especially for the RNA runs, your quality will sort of crater after 48 hours. And this is a sign that, okay, all the reads that are going to be high quality are probably already sequenced. And from this point on, you're just sort of burning pores without actually increasing the real quality output. And finally, fragment size, so your N50, how large your reads are. This, as I mentioned before, I typically use like a 10 kb protocol for my genomics runs. So as you can see, 8.8 kb, not too far off, especially because throughout the rest of the library prep after shearing, it's expected that the fragments get a little bit smaller as well. Okay, so for whatever reason, you've decided now that your run is over and you are ready to finish your run. What do? First thing, of course, just click stop run, stop base calling and sequencing on the MinION software. Before doing this, again, I hope that folks can learn from my previous mistakes. You want to make sure that there's enough space left on the system that you're collecting data on. In the past, I have not done this, and it's led to some data being lost and corrupted. which is really disappointing after you've had a really excellent run and you're eager to look at the data that you've collected. So again, I do this on the SCIC core computer. So if you're doing this, like if you decide to acquire your own sequencer, this might be a little bit easier for you, or you might decide to get like a larger storage system, but always make sure that there's enough room before you end the run and transfer. Next, you want to transfer your data from the core computer or whatever you're collecting the data on to wherever you want to do the analysis. So for us, this means transferring from the core computer to our lab share. And we do this through FileZilla, but there are plenty of other ways to do it. On the SCIC website, there's documentation for like how to set this up, and it's fairly simple. But copy errors can occur, so just be sure to double check that everything gets transferred properly. And in addition, you want to make sure that you transfer this off relatively soon after your sequencing run is finished, because you don't want other people, if you're working through the core, accidentally deleting your data. Finally, you can wash your flow cell. I highly recommend doing this after every single run, even if you think, okay, this flow cell has no more pores functional. If you wash your flow cell afterwards, there's a high chance that you actually recover some pores and you get to reuse your flow cell. I've reused flow cells as many as like 3 or 4 times. It's a fairly simple process. I won't go over the flow cell washing protocol. I don't think it's all too interesting. But yeah, so you can either rerun a library right away, or you can store your flow cell and use it some other time. I believe that flow cells generally have a 3-month expiration date or shelf life. Yeah, so with that, let's talk a little bit about the data analysis pipeline that my lab uses and some practical applications for these sequencing runs. So for us, we decided to do sort of a custom data analysis pipeline. So the sequencer will spit out these pod5 files. Even though they're base called in real time, there are different base calling like algorithms. Some of them are high, like super high accuracy, which take a little bit longer, but make it seem like you have a higher Q score. So the quality of your reads can sort of get boosted after the fact in that case. Or there are like quicker, lower quality base calling algorithms, which might work for some other people's projects. But because so many of these reads are skipped in real-time base calling, I like to also base call after the fact. And from that, I get BAM files, which I can port into a genome browser such as IGV or some sort of methylation visualization software. We use MethylArtist currently, and I'll show examples of what each of these look like in a moment. I did want to mention, though, I have not personally used this yet, but ONT has this open source analysis platform called Epitome. Hopefully I'm pronouncing that correctly. And this is for base calling, alignment and assembly, variant calling, and a ton of other things. So if you're interested in doing this and don't feel like there's a need to develop your own like customized pipeline, most things that you could probably want to do or imagine doing are available through this open source platform. Okay, yeah, so I wanted to really briefly touch on a few examples of how this sequencing technology has helped us with our research and is continuing to help us in our research. So I've been studying this syndrome called Angelman syndrome, which is a genomic imprinting disorder. So for individuals without Angelman syndrome, this gene called UBE3A, which is a ubiquitin E3 ligase, is imprinted in neurons. So only the paternal allele is active and the maternal allele is epigenetically silenced. Whereas in pretty much all other tissue types besides neurons, both alleles are expressed. Somebody with Angelman syndrome has neurons that have completely terminated expression of UBE3A. So for our system, we were curious to see if we could produce iPSC-derived neurons that recapitulate this imprinting pattern, this monoallelic expression of UBE3A. So this is the UBE3A locus ported into IGV. Up top, you see this is the neuronal DNA. Each of these horizontal gray bars is a read from my sequencing run. And up here, you see this is just like the reference genome. So each of these differentially colored vertical lines represents a variant from the reference genome. As you can see, there are a couple variants here that in the DNA and RNA are present pretty much homozygously in every single read. However, there are other ones where every single RNA read has this variant present across every single read, but the DNA reads, every single read is present in the RNA, but the DNA reads, only about 50% of them have this variant expressed. So this is a really good sign that although there is a variant that is heterozygous in the DNA, only one of those alleles is being expressed in the RNA in this locus. So this is a sign that validates some of our previous experiments, such as nascent RNA fish, showing that we have recapitulated, hopefully, this epigenetic silencing. Additionally, we wanted to see, well, we noticed that in this same locus, there seems to be a transcriptional stop point in the iPSC RNA, where after this point and between these 2 points, there are no reads being picked up. However, in this same exact position for the neuronal RNA, which is derived from these iPSCs, we see that this boundary is not really recapitulated. Well, there's several reads that are cut off at this point, but there are tons of reads in between these 2 points, which are being expressed. And so we wanted to see, okay, well, is this transcriptional boundary due to some sort of differential methylation between iPSCs and neurons? So if you see this boundary here on the left, is this boundary here, it's just a slightly different view. So this is iPSC RNA, and these are methyl artist plots that we generated for this same locus, so it tracks 1-to-1. As you can see, there isn't a ton of differential methylation. Each of these black dots here represents a point of cytosine methylation. And there really isn't a ton of difference between these major cytosine methylation coordinates between iPSCs and neurons, which indicated to us that there must be something else happening here that is causing this transcriptional boundary, other than at least cytosine methylation. So even though it wasn't technically a positive result, we would not have been able to glean this nearly as easily without doing the nanopore sequencing. And by the way, this is just one locus, but you get whole genome epigenetics, like the entire genome profile for this, all in the course of like a 2 or 3 day sequencing run, which I think is a really powerful thing. And with that, thank you all so much for listening to my talk and being here today. Thank you.
Thank you very much.
Yeah. Did you folks want to?
Justin, are there any questions for Justin or no?
Oh yeah, go ahead. Yeah.
So can you just comment on how much DNA you need to start with?
For
And
sure.
how many samples you can prepare this in a time? I don't know if you do large numbers at once, but what would be a practical number of samples that?
Yeah, definitely. So there are certain the ONT documentation provides the exact numbers, but I believe that for DNA, at least with the protocol that I've been using, I think it's 1 microgram of DNA starting out. So not a ton. Like if you're extracting from a plate of fibroblasts or iPSCs, you can usually get that from just like a 10 cm plate pretty easily. And then your second question, like how many samples prepped at one time? So I don't do barcoding personally, at least not yet. ONT does offer this, so you can prep.
We can multiplex up to 96 samples. We're looking into 384 for our microbial applications. But right now we offer up to 96, and we can help you determine what's like an appropriate amount of barcode input. So anywhere from 1 to 96. And then we can do lower inputs. If that's useful, I understand like a microgram isn't a lot from cell culture, but it's a lot from other samples. So we can go as low as like 250 kilograms. Just with some tweaking. And I understand that might still be a lot. The reason that it's so much is because a lot of these protocols avoid PCR so that we can preserve that native DNA. We can, of course, do a PCR step, and then the input, is 10 nanograms, 50 nanograms. But if we want to avoid PCR, then yeah, we are gonna probably ask about 250 minimum.
Yeah,
And then just one more.
sure.
But have you had any experience with, programming your flow cell so it only accepts certain types of DNA across the genome?
Oh, yeah, that's called adaptive sampling. Yeah, so again, I haven't personally done that just because I've been more interested in like the whole genome sequencing. But we have looked into that. The only thing is I can usually get like a really high, like a 30x coverage output within like a 2 or 3 day sequencing run without using adaptive sampling. Of course, the coverage is going to be varied like across the genome. But generally speaking, like I haven't really had the need for it. But if you're like really interested in getting like a huge depth of coverage in just like one particular locus, that could be a really useful thing, especially if you're barcoding. I think that'd be useful. But I think that they'll have more to say about it. Yeah. Any other questions from anybody?
I have a question. Can you hear me?
Yes, I can hear you.
First of all, I enjoyed the seminar. It's all new to me, so I have a very naive question concerning cost. What is the cost comparison between doing this work in your own lab versus sending the DNA or RNA or both to a company and having the company do the work?
Yeah, thank you. That's a great question. So I'm not actually aware of how much it would cost to send out to a different company. Maybe you both would have a better perspective on that. I can say that for doing it yourself, a pack of 4 flow cells costs, I want to say roughly like $4,000. The reagents, like the kits have varying costs, but they're within like the 700 to 1,000 range, I would say. But then again, you can reuse your flow cells several times over. So each sequencing run is not nearly that cost. But I think for the output that you expect from each run, I've found that it's extremely worthwhile. But I'm sorry, I can't really give like a fair comparison since I haven't done the sending out for others to sequence for me.
Okay, thank you.
Sure.
I would just speak on that. So it depends on the core and it depends on the service, right? So sometimes there are like upcharges depending on like, whatever the service you don't do. And then included with that is the reagents and everything. But as you see, you're doing it yourself, so you just.
Yeah,
Yeah.
the model that for T-runs is quite nice, right?
Yeah.
Because you have more control and
Right.
I think it is a little bit more accessible cost-wise than having to go full service or provider. So I think that's definitely a benefit compared to a send out to.
Off the top of my head, I think the genome score is about either 1 to 2K per lane, and I'm not sure. I think you can opt in for the but I believe that is the default. So I think it's about average out to the 2 options.
Yeah, that makes sense. One other point that I wanted to make about your first question, regarding like the barcoding and how many samples can you run per sequencing run. I remembered that the Promethean at least has 2 ports where you can plug in 2 separate flow cells. So you can, even if you're not barcoding, you can run multiple samples at one time, which I generally like to do because it just, you can prep multiple libraries at the same time, you can sequence multiple samples at the same time, and it cuts down the overall time and cost for sure to do it that way.
So you run 1 sample per flow cell.
For the
Yes.
human genome, we
For
would
human
recommend to hit 30x coverage. We would recommend for a whole human genome, 1 sample per flow cell. We are pushing our yields higher and higher, so we would like to get to the point of 2 samples per flow cell for whole genome. But then with adaptive sampling, you can do a